everybody! Welcome to our first lecture presentation of the semester, Cells of the Nervous System. This is based on your CALIT textbook, Chapter 1 material. I recommend as you view the lecture presentation that you have the slides in front of you so that you can take notes in the notes section below each slide on any comments that I make that aren't contained on the slides. And you can also have it in front of you during videos so that you can take notes on the relevant points. That way, you'll have the slides document available to you when it's time to work on assignments or study for quizzes and exams. Similarly, I recommend having the interactive study guide available. I personally would complete it as I'm viewing the lecture presentation and the videos. The interactive study guide provides a snapshot of the most important information for that material, and it will predominantly be the material on your Kahoot quizzes as well as on your exams. With that said, I do want to note that your Quizlet flashcards provide the answers to the interactive study guide questions that have specific answers. Questions that ask about what you gained from watching a video or that are more subjective in nature are not included in the Quizlet flashcards. They are not on the Kahoot quizzes, but they will be on your exams. So it is very important that you fill out those sections and that you study accordingly. In terms of the videos, there are two ways to go about integrating the lecture presentation and the video material. One option is to watch the lecture presentation in its entirety and then watch each of the videos to reinforce your learning. The other option is to intermix the lecture presentation with the videos. I'll do my best on the relevant slides to point out that there is a video on that topic. You could then pause the recording, go and view that video, and then return back to the lecture presentation. So this would be more similar to sitting in class, watching and listening to some lecture material and then viewing a video and then going back to the lecture material. You might consider trying out both strategies and seeing which one works better for you. Perhaps week 1, you watch the lecture presentation all the way through, and perhaps in week 2, you choose to try going back and forth and see if that better suits your learning. There's really no right answer, there's just the best answer for you. We'll start off by discussing the typical animal cell. I do want to note for those of you who would like to try the strategy of intermixing the videos with the lecture presentation, you might consider pausing here to watch the video Introduction to the Nervous System. So the information on this slide applies to all animal cells except mammalian red blood cells. So all of these cells have what's known as a cell membrane or a plasma membrane, which is the surface of the cell that helps separate the inside of the cell from the outside environment that surrounds the cell. The plasma membrane contains protein channels that help permit a controlled flow across the membrane. The nucleus contains the genetic material, such as the chromosomes for the cell. Mitochondrion, which is the singular of mitochondria, perform metabolic activities. The activity of mitochondria affect energy levels. Ribosomes help synthesize new protein molecules. They can attach freely, or they can attach to the endoplasmic reticulum, which is a network of thin tubes that help transport proteins. There are many different types of cells, but one that is particularly important for this class is the neuron. Neuron is basically just the scientific term for a nerve cell. And while they typically are presented to look as though they are all the same, they actually vary in size, in shape, and in function. We're going to start by talking about two ways of looking at neurons, or two different types of neurons, and those are motor neurons and sensory neurons. And I want to bring up the terms efferent and afferent. Some of you might be familiar with these terms, but some of you might not be. So all cells can be efferent and afferent. The term refers to whether the information is going away from a structure or going towards a structure. So for the example of motor neurons, Motor neurons are considered efferent to the central nervous system. The mnemonic you can use to help remember that is efferent is to exit. Right? So motor neurons have their soma, or their cell body, in the spinal cord. When it receives excitation from other neurons, it sends impulses along the axon, which is basically the tail that extends from the soma, towards the muscles and glands. So we would say that that information is exiting the central nervous system and headed towards the muscles and glands. In contrast, sensory neurons are afferent to the central nervous system. That's because the information is traveling towards the central nervous system. And so the mnemonic for that is afferent is to arrive. Sensory neurons are sensitive at one end to a particular type of stimulation, and that type of stimulation varies across neurons. So for example, close to your skin, there are nerve endings that are sensitive to touch, to heat, to pain. There are also neurons that are sensitive to light or to sound. The information then travels towards the central nervous system to be processed. Interneurons are a smaller, specialized type of neuron, and they're also referred to as intrinsic neurons. To be an interneuron, the entire neuron has to be contained within a single brain structure. So that means that all of the dendrites and axons, all of the parts that extend from the soma, and we'll talk about those parts in just a little bit, have to be contained within one singular structure. So next we'll talk about the parts of a neuron. For those of you who would like to pause the recording to watch the video, you might consider going to watch the optional video, Neurons Forming Connections. I made this video optional because it does require Facebook. I hope that many of you will be able to access it because it is a really neat way of seeing neurons forming connections in real time. So before we go through all the parts of the neuron, I do want to note that not all neurons have every part. Some neurons only have a soma. So we're going to start with dendrites. Dendrites are branching fibers that gradually narrow as they get further and further away from the soma. You can think of them as being like tree branches that extend from the soma, which is basically the trunk of the tree as far as the dendrites are concerned. Dendrites are lined with synaptic receptors, and those are going to be very important when we start to talk about cellular communication. Dendrites also have what are known as dendritic spines that are basically little protuberances that extend from the dendrites themselves. So if you look at the picture in the bottom right corner of the slide, you'll see that is a soma with the nucleus and mitochondria, and extending out from that round soma are the dendrites. In the top image, you'll see the lighter kind of yellowy orange color. Those are the dendrites. The darker orange colors are the dendritic spines. So you can see that there are these extensions that kind of grow out of the dendrite. And those are really important because they increase the surface area of the dendrite and allow for more receptors to be placed there. This is going to be a really important concept when we talk about the pathophysiology of depression in Unit 2. One of the leading theories of the kind of biological aspect of depression is that these dendritic spines atrophy, which means that they retract into the dendrites themselves. And in doing so, the individual loses surface area for receptors. The next part of the soma the next part of the neuron that I've already touched on a little bit is the soma. And again, the soma is the cell body. It contains the genetic material, the nucleus, the ribosomes, the mitochondria for the cell. It is the site of the majority of the metabolic action, and like the dendrites, it often contains synaptic receptors on its surface as well. The axon is a thin fiber, and unlike the dendrites that get narrower as they get further away from the soma, the axon is a constant diameter from where it attaches to the soma all the way to where it ends at the terminal button. The axon conveys impulses towards other neurons, organs, and muscles. And it always does this in the same direction, which is away from the soma. The myelin sheath is an insulating material that surrounds some axons. Not all axons are myelinated. Those of you who are familiar with the disease multiple sclerosis might know that multiple sclerosis is a condition caused by degeneration of the myelin sheath, otherwise known as demyelination. The nodes of Ranvier are the interruptions in the myelin sheath. You could also think of them as being gaps between sections of myelin. That will be important when we talk about action potentials later on. The presynaptic terminal has a lot of different names. Some people refer to it as the presynaptic terminal. Some refer to it as the terminal button. You might also hear synaptic button or synaptic bouton. They all mean the swelling at the very tail end of the axon, which is the location where the majority of neurotransmitters are released. It is the furthest point away from the soma and dendrites. An interesting fact about axons is they can be more than 1 meter long. And I want you to think for a minute, why would you have an axon that's that long? The answer is that you need to be able to reach the areas that are furthest away from your spinal cord. So for example, your feet. Now, those of you who are taking this course online don't know that I'm relatively short. I'm 5'1". I don't need to have 1 meter long axon. But, for example, Shaq would need to have a really long axon to reach from his spinal cord all the way down to his feet. The next type of cell that we'll talk about are known as glial cells. Glial cells are also referred to as glia and as neuroglia. There are many different types of glial cells, and we'll just touch on a few of them. First, astrocytes. Astrocytes have a lot of important functions in the body. One of them is that they wrap around synaptic terminals of axons. They help take up ions and various types of transmitters that are released by axons and then release them back. So they're actually active partners with neurons. They actually help synchronize neurons, which enables waves of messages. Now, why would it be important for neurons to synchronize themselves? One example is breathing. It's really important when you take a breath that your right side of your lung and your left side of your lung are relatively equal in their work. Astrocytes also help support nutrient needs. They do this by dilating blood vessels, which helps bring more nutrients in. Next are microglia. Microglia basically are the scavengers or the janitors of the central nervous system. They proliferate after brain injury, and they help remove dead and damaged neurons. They also contribute to learning by removing weak synapses. Radial glia are another type of glial cell that aren't noted on this slide, but I do want to mention briefly since they are in the picture. Radial glia guide migration of neurons during embryonic development. They then differentiate into neurons, astrocytes, and oligodendrocytes, which is what I'm going to talk about next. Oligodendrocytes help with myelination in the central nervous system. So they actually myelinate axons in neurons in the central nervous system. And you can see that in the middle of the picture. Similarly, Schwann cells also perform myelinating roles, but they do so in the peripheral nervous system. And you can see them a little further up in the image, kind of shaded in a purple color. If you're interested to know what this looks like, I recommend the video 2-Minute Neuroscience BloodBrain Barrier. We'll be talking about the blood-brain barrier on the next slide, and it gives some helpful images in about 2 minutes. You can find this video on YouTube, again, by searching 2-Minute Neuroscience Blood-Brain Barrier. I didn't include it in the required videos because it is so short, and it is not actually a requirement that you see it visually, though I do think it would be helpful, particularly for those of you who really thrive on being able to see things come to life. As I mentioned on the last slide, we're now going to talk about the blood-brain barrier. Just like it sounds, the blood-brain barrier protects the brain by keeping it relatively separate from the rest of the bloodstream. The blood-brain barrier is important for keeping out toxins, bacteria, viruses, and all kinds of other harmful substances. It's important that we go the extra mile to protect the brain because unlike skin and red blood cells, neurons do not regenerate. And it's only in very, very few brain regions that neurons are able to completely undergo what is known as neurogenesis, which is the creation of new neurons. So the blood-brain barrier does help prevent the crossing of some harmful chemicals and substances, similar to the placental barrier. However, it also keeps out some useful chemicals as well. This impacts various types of medications. So for example, the medication lithium does not cross the blood-brain barrier easily, and therefore you have to use a higher dose for enough of it to be available by the time it is able to cross. Lithium crosses by active transport, which is required for many chemicals to cross the blood-brain barrier. If you look at the picture, you'll notice there are green arrows and purple arrows. The green arrows refer to substances that cross passively, which means that they are able to basically slide through the blood-brain barrier without any extra help. The purple arrows refer to active transport. Active transport is a protein-mediated process where basically a transporter takes that substance across the blood-brain barrier because it couldn't otherwise cross. Glucose is the only nutrient that easily crosses the blood-brain barrier. A really interesting fact is that the brain is only 2% of our body weight, but uses 20% of our oxygen and 25% of our glucose. So we need to get a lot of glucose into the brain to have sufficient quantities. The body needs thiamine, which is vitamin B1, to use glucose. Those of you who have learned about Korsakoff's syndrome, which is a result of heavy alcohol abuse over many years, might be familiar with thiamine. Korsakoff's is caused by cell death due to insufficient thiamine, which then leads to memory impairment. The good news is that small uncharged molecules like oxygen and highly fat-soluble molecules like vitamin D cross freely, otherwise known as cross passively. As I mentioned, active transport is required for many other substances. Now, the area postrema is a region of the blood-brain barrier that is intentionally weaker than many other areas of the blood-brain barrier. And I want you to ask yourself for a moment, why would there be an intentionally weak area of the blood-brain barrier? Why would that exist? The answer is that the area postrema, which is also known as the chemoreceptive zone, is an area of the brain that is tasked with kind of checking in on the rest of the bloodstream. So for example, if you had food poisoning, the response that your body undergoes in order to protect you is to induce vomiting. It is your brain that triggers that vomiting. If no region of your brain could tap into the rest of your body, how would it know to trigger that response? So the area postrema is intentionally weak as a way for your brain to ascertain what's going on in the rest of your body. Those of you who watched the blood-brain barrier video also were introduced to another weaker region of the blood-brain barrier, which is around the pituitary gland. The reason for that intentionally weak area is that the pituitary gland needs to be able to drop hormones into general circulation in the bloodstream. We're getting closer to our discussion of action potentials, but first we need to learn about cell membrane resting potentials. The cell membrane is composed of what's known as a phospholipid bilayer. Protein molecules are embedded in this phospholipid bilayer and allow the passage of certain chemicals. And you can see that in the image. At rest, the cell membrane maintains its electrical gradient, and we call that polarized. So at rest, the neuron is polarized. That means that the electrical potential inside of the cell is more negative than outside of the cell. The difference in voltage varies across neurons. but the resting potential at average is typically around -70 millivolts, though it could go anywhere from about -40 to -90 millivolts. Messages in a neuron develop from disturbances in this resting potential. Again, at rest, the inside of the cell is significantly more negative than the outside of the cell. And this is particularly due to negatively charged proteins that are predominantly contained inside the cell. Surrounding the cell in the extracellular fluid are many sodium ions that are actually 10 times more concentrated outside the cell than inside the cell. So you can imagine sodium, which is Na+, it is a positive ion, in heavy concentration outside the cell, and many negatively charged proteins inside the cell. That predominantly explains the difference between the very positive extracellular fluid and the very negative intracellular environment. Selective permeability helps prevent unwanted depolarization. Depolarization is when the inside of the cell becomes more positive and gets closer and closer to initiating an action potential that we'll talk about soon. First, I want to mention the sodium-potassium pump. The sodium-potassium pump actively transports 3 sodium ions out for every 2 potassium ions in. This helps maintain that high concentration of sodium outside the cell. There's also a high concentration of potassium inside the cell, but again, those negatively charged protein ions are in higher concentration than potassium, and that is why the inside of the cell is so negative. Sodium primarily stays out when the channel is closed, but some potassium is able to leak out even when the channel is partially closed. There are 2 types of gradients that are important to understand, the electrical gradient and the concentration gradient. The electrical gradient explains that sodium, being positive, is attracted to the negative inside of the cell. So the electrical gradient in general is that ions are attracted to their opposite charge. The concentration gradient explains why sodium is attracted to lower concentrations. So for sodium, the electrical gradient and the concentration gradient are influencing it towards the inside of the cell. It's heavily motivated to get where it is more negative and to get where there is less sodium. Sodium is ready to enter, but at rest, it can't leave. It's ready to go, but the channels are closed. Potassium is attracted in by the electrical gradient, but it is attracted out by the concentration gradient. Therefore, the sodium-potassium pump counteracts the potassium that leaks out when those channels are partially closed. Okay, moving on to action potentials. So we talked about the term polarized, which refers to the fact that the neuron is at rest. So 2 other important terms to know are hyperpolarized and depolarized. Hyperpolarization refers to when the inside of the cell becomes even more polarized, which means even more negative in comparison to the positive external cellular environment. We would say that that neuron is inhibited because it is further away from initiating an action potential. Depolarization is the opposite. Depolarization refers to less polarization, which means that the internal environment is getting increasingly more positive. We also would refer to that neuron as excited. So the more negative the internal environment of the cell, the more inhibited that cell is. The more positive the internal environment of the cell, the more excited that neuron is. If you look at the picture, you'll see those terms, hyperpolarization and depolarization. You'll see that there is the resting potential. That is, again, where the cell is at rest and is polarized. That's that black dotted line at the beginning of the graph. The line right above that, it's a red dotted line that says threshold. That is what's known as the threshold of excitation. When there is sufficient depolarization or excitation, as that cell becomes increasingly positive inside, it gets closer and closer to hitting that threshold. So let's look through this graph. We start at resting potential. Then you'll notice the black line dips down below the resting potential. This is hyperpolarization. It is inhibited. Then you'll notice it goes back to resting potential for a little bit. Then it drops down even further than it did before. So it is even more inhibited than during that first IPSP. IPSP is another term that refers to the inhibition, right? So it is called an inhibitory postsynaptic potential. In contrast, you'll notice after it goes back to resting potential, there's this little hill. It has not quite reached the threshold, but it is above the resting potential. These are referred to as EPSPs, which stands for excitatory postsynaptic potential. It is many EPSPs that will eventually get the cell to reach that threshold of excitation. So you'll notice it goes back down to the resting potential, and then on this next EPSP, it hits that red dotted line, and at that point initiates the action potential. And you'll notice the action potential goes to the same peak on each of those action potentials. And that's because we refer to action potentials as all or nothing. If that threshold is hit, it goes to the peak of excitation every time. It doesn't matter if it was barely to the threshold or if it fully exceeded the threshold. The intensity of the action potential is always consistent. This is known as the all or none law. So, when the cell is depolarized, voltage-gated channels open, which allows that sodium that we talked about earlier that's ready to rush into the cell. It allows it to come flooding in, and it allows potassium to exit the cell, but it exits at a lower rate because it's not as motivated. At the peak of the action potential, sodium channels are snap closed, but potassium channels remain open. This initiates what's known as the refractory period, and we'll talk about that in more detail on the next slide. Potassium helps to repolarize and potentially to hyperpolarize the cell, and then the sodium-potassium pump fully restores the resting potential. For those of you who would like to see this in action, I recommend pausing the recording and watching the video, Lights, Camera, Action Potentials, located in your weekly materials. I want to also mention something interesting about sodium. A buildup of sodium in the cell can actually be toxic, and it's one of the reasons why strokes, as well as certain drugs of abuse, can cause toxic effects. Another interesting fact about sodium is that local anesthetic drugs like Novocaine work by blocking sodium ions from entering cells. So the receptors would transmit pain, but the axons can't initiate the action potentials that are needed to transmit that information to the brain. So you don't experience pain, even though the receptors are bringing in sensations of pain. So this provides another way of visualizing the stages of the action potential and the refractory periods. The interactive study guide provided in this week's materials also presents this graph. So as we're going through this slide, I recommend filling in the stages. Starting with the far left of the chart. So you'll see the neuron starts about negative 70 millivolts at its resting potential. So at this point, the cell is polarized. In stage 2, you'll notice that the cell undergoes an excitatory postsynaptic potential, a local potential, an EPSP, that depolarizes the cell to the threshold. That threshold, at the border between stage 2 and stage 3, is the beginning of the action potential. Stage 3 is the rising phase of the action potential. It is the phase from the threshold of excitation to the peak of the action potential. So at the border between stage 3 and stage 4, you're at the peak of the action potential. From here, stage 4 represents the refractory period. The first part of stage 4 is known as the absolute refractory period. So this is the first part of the refractory period, where the cell cannot produce an action potential under any circumstances. The second part of stage 4 is the relative refractory period. Here, an action potential is possible, but it takes a much stronger stimulus to trigger it. That should make some logical sense. At this point, the cell is hyperpolarized. You can see that because the red line that represents the activity of that neuron, you can see it dips below the resting potential. So it is hyperpolarized at that point. That makes this gap between where it's at and the threshold larger than it is between a resting potential and that threshold. So the cell would have to be even more stimulated to get it to the threshold of excitation. The relative refractory period depends on the fact that sodium channels are closed and potassium is leaving the cell at a faster than usual rate. To give you an idea of timing, the absolute refractory period usually lasts about 1 millisecond, and the relative refractory period usually lasts between 2 and 4 milliseconds. The refractory period explains why action potentials cannot travel backwards. They always go in one direction, from the soma towards the terminal buttons, so down the axon, away from the soma. And again, as I mentioned earlier, the sodium-potassium pump is what fully restores the neuron to its resting potential so that it is polarized again. All right, our last slide for this lecture presentation, the propagation of the action potential. So as I've mentioned, action potentials always travel in one direction, and that direction is down the axon, away from the soma, and towards the terminal button. As I mentioned, myelin insulates the axon on some neurons and speeds up the action potential. So you can think of the myelin around the axon being similar to the coating around an electrical wire. It helps protect that wire, and it also helps to facilitate a more efficient route of communication so that the information can travel more quickly. It doesn't degrade as it goes. That coating really protects it. The nodes of Ranvier are those gaps between myelin, and you can see it in both of the images. So in the top image, the purple represents the myelin, and that yellow color represents the axon, and you can see that the myelin has little breaks between it. And it looks like a link of sausages. You can also see it in the bottom picture. The nodes of Ranvier are the circular portions in between the longer sections of myelinated axon. The nodes of Ranvier expose the axon to extracellular fluid because those are the areas that are not myelinated. Action potentials start at the first node of Ranvier, as close to the soma as possible. From there, action potentials on axons that are myelinated undergo what's known as saltatory conduction. Saltatory conduction basically refers to the fact that the action potential looks like it jumps from node to node. That's because it basically refuels at each node. When it goes through those myelinated sections, it's basically going through a rapid tunnel. There's no place for those ions to escape or for them to come in. When it gets exposed to that node of Ranvier, there's a chance then for the action potential to regenerate itself by allowing more sodium into the cell. As I mentioned before, multiple sclerosis is a condition caused by myelin degeneration. Unmyelinated axons differ from those that were previously myelinated and lost their myelin. They don't have sodium channels where myelin used to be, which then leads the action potential to die out, which means that the communication that the action potential is trying to facilitate ultimately is unable to reach the next neuron. Next time, we're going to learn about how neurotransmitters and hormones facilitate this cellular communication. So action potentials are what gets the energy down to the terminal button. But the next step is how do these neurotransmitters and hormones get released into the extracellular fluid? And then what do they do from there? How do they go and impact their neighboring cells? That's where we'll pick up next time. And until then, if you have any questions, comments, or concerns, please come to office hours or reach out to me.